So picture trying to repair like a really complex piece of heavy machinery that's failing dangerously.
Okay.
But the specific part causing the problem is completely smooth and gray.
Yeah.
I mean, there are no serial numbers, no exposed gears, no warning lights. You literally can't tell what it is or how it connects to the rest of the machine.
Right. It just leaves you entirely in the dark.
Exactly. You just know it's systematically destroying the engine from the inside out.
And you can't safely remove it or fix it because, well, any tool you use would just be a total blind guess.
Yeah, and that is the exact terrifying reality pathologists face when they encounter what's called a sinonasal undifferentiated carcinoma, or a SNUC for short.
Yeah, these are extremely rare, really aggressive cancers that form deep in the nasal cavity.
And under a microscope, they lack any defining visual features, right?
Yeah.
Like they don't look like skin cells, they don't look like gland cells. They are just a featureless, undifferentiated mass.
Which is why, historically, they've been so incredibly difficult to treat. I mean, when doctors can't identify a tumor's biological origin, they're forced to rely on generalized, highly toxic approaches.
Like standard chemotherapy.
Exactly. And unfortunately, for these specific tumors, those blunt force treatments frequently fail.
But that is exactly what we're unpacking in today's deep dive. We were looking at a groundbreaking study that found a way to look completely past that featureless exterior.
Yeah, the researchers essentially figured out how to read the genetic diaries of individual microscopic tumor cells to map out their hidden vulnerabilities.
So for you listening, here's our mission today. The source material we have is a highly detailed molecular analysis. The research team took 12 preserved tumor samples from the sinonasal cavity.
Right, and half of these tumors carried a mutation in a gene called IDH2, and the other half had a mutation in a gene called SESMAR-RC4.
Right.
And to really understand these tumors, they used a technique called single nucleus RNA sequencing to hunt for potential targeted treatments.
So just to recap the intro here, we're exploring rare, featureless nasal cancers. We're using high-tech single-cell sequencing. And the goal is to find drug targets. I mean, if a tumor has no visible defining features, how do you even begin to attack it? Let's find out.
It's a great question. And before we get to the specific vulnerabilities, we really need to talk about how they even pulled this off.
Yeah, because the raw material we're working with sounds like an absolute nightmare for a geneticist. Like, these were not fresh tissue biopsies.
Oh, far from it. The researchers were working with archival samples, specifically FFPE tissue, that stands for formalin-fixed, paraffin-embedded.
Which is basically the medical equivalent of an insect trapped in amber.
That's actually a perfect way to describe it.
I mean, the tissue is dunked in chemicals and encased in hard wax, so it can sit on a hospital storage shelf for decades without rotting. It's fantastic for preservation, but it essentially petrifies the sample.
Yeah, the chemical fixing process cross-links all the proteins and nucleic acids inside the cell. It tangles everything into this dense knot, making it notoriously difficult to extract high-quality genetic material.
Because older sequencing methods simply couldn't handle that.
No, they couldn't. But the breakthrough here is the successful application of single-nucleus RNA sequencing on this petrified tissue. They actually managed to extract and isolate 59,612 individual nuclei from these samples.
That is just wild. Nearly 60,000 individual profiles. I want to give you, the listener, a way to visualize why isolating single nuclei is such a massive deal.
Go for it.
Think of traditional genetic sequencing, like bulk sequencing, as placing a single microphone in a room with a choir of 60,000 people. You hit record, and you get a massive wall of sound.
Right, a complete blend.
Yeah. If you're trying to figure out which specific singer is off-key or singing a completely different song, it's nearly impossible. You just hear the average of the whole room.
Exactly. Bulk sequencing blends the cancer cells, the immune cells, the blood vessels, the structural tissue, all into one generic data point. But single nucleus sequencing changes the paradigm entirely.
It's like pulling all 59,612 singers out of that choir.
Placing them into their own individual soundproof booths and interviewing them one by one.
Okay, so they're looking at the RNA of each nucleus, the messenger molecules that tell us exactly which genes that specific cell is turned on or off. Wait, I have questions.
Sure.
If this tumor is a featureless, undifferentiated gray mass, how do you know which singer in the soundproof booth is the malicious cancer cell, and which is just a normal, healthy nasal cell caught in the crossfire?
Well, you look for structural chaos. Healthy cells maintain a very strict, highly organized set of chromosomes, but cancer cells completely lose control of their replication machinery. They suffer from severe genomic instability.
Genomic vandalism. They basically start tearing up their own blueprints.
Yes, and that results in something called copy number variations, or CNVs. Because the cell's division process is broken, it will accidentally duplicate massive chunks of chromosomes or just delete entire critical sections altogether.
Oh, wow. So they just look for the messy cells.
Exactly. By analyzing the RNA data, the algorithms can infer these large-scale chromosomal changes. If a single nucleus showed a chaotic landscape of massive duplications and deletions, the researchers flagged it as a malignant cancer cell.
And if its genome looks stable?
Then it was categorized as a non-malignant cell in the tumor's ecosystem.
Which is fascinating because it really is an ecosystem. Even in these featureless masses, they mapped 16 different types of normal cells interacting with the cancer, like T cells, fibroblasts, blood vessel cells.
Yeah, it's incredibly complex.
So just to do a quick mini summary for you listening. Step 1, extract nuclei from preserved amber-like tissue. Step 2, sequence them individually in soundproof booths. Step 3, use chaotic chromosomal changes, those CNVs, to spot the bad guys.
Perfect summary.
So now that we've isolated the cancer cells, what are they actually doing inside that tumor?
Well, this is where the narrative shifts from looking at a single enemy to looking at a complex army. The researchers found profound intratumoral heterogeneity.
Meaning the tumor is not a monolithic blob.
Right. It's made up of distinct, highly specialized subpopulations of cells that are running completely different biological programs.
So looking at the data, it seems like these cancer cells fall into a few distinct behavioral profiles. Let's trace these out. You have one massive group of cells that are entirely focused on energy consumption and rapid proliferation. Like they're running incredibly hot.
Yeah, their genetic pathways show heavy enrichment for oxidative phosphorylation and metabolic activity. They also show strong activation of MYC targets.
MYC, that's an oncogene, right?
Yes. MYC functions essentially as a cellular gas pedal.
Okay, so let's call this group the engine. Just churning, burning fuel, forcing the mass to grow. But then right next to them, in the same nasal tumor, you have this second group of cells acting completely bizarre.
They really are. They're activating pathways usually reserved for nervous system development.
Wait, like brain cells?
Sort of. They exhibit a strong neuroectodermal identity, expressing genes that typically guide the growth and differentiation of neurons. But what makes this subpopulation so unstable is that they're simultaneously showing active signaling for apoptosis.
Which is programmed cell death.
Exactly.
So let's call them the confused neuron. They are trying to grow like brain cells, but their internal alarms are screaming at them to self-destruct.
It creates a highly erratic, stressed cell state. And crucially, this specific neuroendocrine-like behavior was found predominantly in the tumors that carried the SMARCA4 mutation.
Okay, so SMARCA4 tumors lean heavily on these confused neurons. Got it. What else is in the army?
Well, you have a third distinct group that specializes in weathering the harsh conditions of the tumor microenvironment.
Because rapidly growing tumors often outpace their own blood supply, right?
Right, leading to areas of low oxygen, high acidity, and toxic buildup. This group of cells responds by pumping out massive amounts of heat shock factors and stress proteins.
Which act like cellular first responders. When the cell is under extreme duress from, toxins or lack of nutrients, these proteins rush in to stabilize the internal structure so the cell doesn't just collapse on itself.
Precisely.
So these are the stress survivors, just bracing against the environment, or honestly, bracing against whatever drugs the doctor throws at them.
And finally, the fourth major behavioral group is driven by a biological process called epithelial mesenchymal transition, EMT. This is a critical hallmark of cancer metastasis.
This is where the cells stop being sticky, right?
Exactly. Normal epithelial cells adhere tightly to their neighbors. During EMT, they lose that adhesion and gain the ability to migrate and invade surrounding healthy tissue,
making them the invaders. And looking at the pathway driving this, it's heavily reliant on a protein called TGF-beta, which is a fascinating molecule because it has a sort of split personality in biology.
Yeah, TGF-beta is the classic double agent of oncology.
How so?
Well, in healthy tissue or very early stage benign growths, TGF-beta actually acts as a strict tumor suppressor. It acts as a brake, telling cells to stop dividing.
But in advanced cancers?
In advanced mutated cancers, the tumor completely rewires that signaling network. Suddenly, TGF-beta becomes a massive promoter of invasion, pushing the cells to spread while simultaneously helping the tumor hide from the immune system.
Wow. So we have engines, confused neurons, stress survivors, and invaders.
Oh,
And wait, we mentioned the SMARCA4 mutated tumors favor those neuron-like cells. What about the IDH2 mutated tumors?
Good catch. They run an entirely different program. The single-cell data showed the IDH2 tumors heavily activate genetic pathways that resemble tuft cells.
Tuft cells?
Yeah, they're specialized sensory cells naturally found in the mucosal linings of the body. So while to the naked eye both tumors are identical gray blobs, at the RNA level, one is attempting to mimic a neuron and the other is attempting to mimic a sensory mucosal cell.
When you step back and look at that whole picture, the sheer terror of this disease becomes so clear. Think about a patient receiving standard chemotherapy. You are fighting a mass that contains engines designed to grow rapidly, confused neurons misfiring, stress survivors biologically adapted to resist toxicity, and invaders actively trying to spread. No wonder a single generic drug fails.
Right. You might successfully kill off the rapidly dividing engine cells, but the stress survivors simply weather the treatment and eventually regenerate the tumor.
So just to pause and recap for our listener, we've got an army with different cell states, engines, neurons, survivors, and invaders. And the overarching mutation, either susMRA or IDH2, dictates their main lineage. So knowing this, how do we target an enemy that has so many different faces?
Well, that's the ultimate goal here. The researchers scoured this RNA data looking for specific proteins that these tumor cells were overproducing compared to normal tissue.
They were looking for a lock they could put a key into.
Essentially, yes. They focused heavily on kinases, which are enzymes that act as on-off switches for various cellular functions. Kinases are notoriously druggable.
Okay, let's start with the IDH2 tumors, the ones mimicking those tough cells. The data shows they heavily overexpress a receptor called KIDAC, which normally promotes cell growth. But I read the methodology closely, and this is where I get a bit confused.
Oh, what part?
So the researchers ran targeted DNA sequencing, and they confirmed there are 0 activating mutations in the KIT gene itself.
Right.
The switch isn't broken, it's not permanently jammed in the on position. So if it's just a normal unmutated gene, why is it a target? Why would blocking it hurt the cancer?
It comes down to a concept called oncogene addiction.
Okay.
In a normal cell, you have a strictly regulated number of these KIT receptors. But these tumor cells are producing massive abnormal quantities of them. Even though the individual receptors aren't structurally mutated, the cell becomes entirely dependent on this overwhelming baseline signaling just to stay alive.
Oh, I see. So they haven't broken the switch, they've just built a million of them, and they desperately need all of them flipped on to maintain their chaotic metabolism.
Precisely. The whole infrastructure of the cell is so warped around this overproduction that if you introduce a drug to suddenly block those KIT receptors, the cell suffers catastrophic withdrawal and basically collapses.
That makes total sense.
Yeah.
And the beauty is we already have drugs that block KIT, right? Like inhibitors currently used for gastrointestinal tumors might actually be repurposed here, assuming clinical trials validate the RNA data.
Exactly.
So what about the SMARCA4 tumors?
They present a different but equally actionable vulnerability. The SMARCA4 tumors, those with the neuron-like cells, massively overproduce a completely different receptor called MET.
And
again, physical testing, like FESH testing, confirmed the gene wasn't amplified or structurally multiplied. The cells were just reading that specific gene over and over, flooding their surface with MET proteins.
And targeting MET opens the door to some truly futuristic medicine. Instead of a systemic chemical bomb that poisons the whole body, overexpression of MET makes the tuner a candidate for an antibody-drug conjugate. An ADC.
Yeah, ADCs represent a massive leap in precision oncology.
They are essentially microscopic smart delivery vehicles. To visualize it, think of modern warfare. The antibody portion is engineered to recognize and bind exclusively to the MET proteins coating the outside of the tumor cell. It acts as a homing beacon.
Right.
And once it physically attaches to the cancer cell, the cell absorbs it, and the ADC releases a highly toxic payload directly into the cell's interior. It destroys the cancer while leaving the surrounding healthy nasal cells relatively untouched.
It's brilliant. It turns the tumor's own overproduction completely against it. And beyond these unique targets kit for IDH2 and MET for SMARCA4, the sequencing revealed a shared weakness across the board.
A shared weakness.
Yeah. Both types of these rare tumors heavily overexpress CDK4.
Which is an enzyme that acts as a critical checkpoint for cell division. So if they're overproducing it to fuel their rapid growth, we could potentially hit them with CDK4 inhibitors.
Exactly. Drugs like palbociclib, which are already widely used and highly effective in certain types of breast cancer, could be brought into play.
Amazing. So let's do a quick recap of the locks and keys we found. For IDH2, we target KIT. For SMART-K4, we target MET with those smart bombs. And for both, we can potentially use CDK4 inhibitors.
Ah, the playbook.
If you were a doctor listening to this, you'd have to decide whether to attack the unique features or the shared ones. But wait, what if the tumor is using its physical environment as a shield?
And that is exactly what this study uncovered next. While identifying these kinase targets is a huge step forward, the researchers found a secondary mechanism that is, honestly, arguably the most sinister discovery in the paper.
The collagen DDR1 axis. This blew my mind, because we tend to think of tumors as just growing passively into empty space. But this shows the cancer actively engineering its environment.
Oh, very actively. Every cell in your body is surrounded by an extracellular matrix. It's a physical scaffolding largely made up of a strong protein called collagen I. To expand, the tumor secretes specialized enzymes that literally chop up that collagen scaffolding.
So it's destroying the walls around it to make room.
It is, but it doesn't just sweep the debris away. The researchers discovered that those cleaved, broken pieces of collagen physically bind to a specific receptor on the surface of the tumor cells.
A receptor called DDR1.
Yes.
So the broken collagen acts like a key fitting perfectly into the tumor's DDR1 lock.
And when that connection is made, it sets off a massive internal alarm bell. The activated DDR1 receptor triggers a complex chain reaction. It activates NF-κB, which then increases the expression of specific stress response proteins like SQ-STM1 and NRF2.
Okay, that's a lot of acronyms. What does that cascade actually do?
Basically, it turbocharges the tumor's metabolism. It directs the cell to go into overdrive, which is exactly what allows those stressed survivor cells we discussed earlier to thrive in such a harsh, nutrient-deprived environment.
It is literally using the destruction of its own environment as a fuel source. And here is the wildest twist in the whole system, listener. If the collagen scaffolding around the tumor remains completely intact and healthy, the DDR1 receptor never activates.
In fact, if the collagen remains intact, the tumor degrades its own DDR1 receptors.
Wow. The tumor is biologically compelled to destroy its environment to power this survival pathway.
It has to. And the researchers didn't just guess this was happening based on the sequencing data. They actually proved it spatially.
Using techniques like RNA-scope and sequential immunofluorescence, right?
Yeah, they literally color-coded the tissue under a microscope. They mapped exactly where the collagen was physically broken and mapped exactly where the DDR1 survival signals were firing inside the cells.
And they lined up.
The alignment was perfect. The tumor's metabolic turbocharge was only happening in the microscopic physical zones where the scaffolding had been destroyed.
So to visualize this, imagine the tumor is a criminal locked inside a sturdy brick building. To survive and grow, it starts knocking down the brick walls. But instead of just escaping through the hole, it wires the broken bricks into its own generator. It uses the destruction to power itself up.
That's a great analogy.
And to make matters worse, all that dense rubble it created forms a physical barricade that keeps the police-like your body's immune system from getting inside.
It's an incredibly robust adaptation, but like all adaptations, once you understand the mechanism, it becomes a glaring vulnerability.
Right. If you introduce a targeted drug, say like dasatinib, to block that DDR1 receptor, you break the circuit.
Exactly. The broken collagen can no longer plug in. The tumor loses its metabolic power source, and that dense protective barricade of rubble begins to fail.
Which really brings us to the ultimate takeaway of this deep dive. Let's recap. We went from a featureless gray blob to extracting single cells, identifying their unique behaviors, mapping their kinase vulnerabilities, and discovering how they use broken collagen to fuel themselves.
Yeah, for decades, a diagnosis of a featureless, undifferentiated tumor was essentially a dead end. We had no specific weapons because we couldn't see any specific targets.
But this research fundamentally proves that just because a cancer looks undifferentiated under a microscope doesn't mean its biology is unknowable.
Right. By isolating 60,000 individual cells and reading their genetic output, we can move away from generic failing treatments. We can match a specific KIT inhibitor, a MET-targeted delivery vehicle, or a DDR1 blocker to the exact biological state of a patient's tumor.
It is the absolute blueprint for true precision medicine. But as we wrap up, there's one final detail from the research that hints at what the next chapter of this fight might look like, and I want to leave you, the listener, with this thought.
That's a very compelling point.
We talked about that dense rubble keeping the immune system out. Well, the researchers noted that T cells, the assassins of your immune system, were actually present in the tissue surrounding the tumor. They were actively trying to communicate with the cancer cells through an immunosuppressive receptor called CD96.
But despite that presence, the tumors remained immunologically cold. The immune cells were locked out, unable to penetrate the tumor bed and execute their attack.
The barricade held. But it leaves you with a fascinating question to mull over as you go about your day. If we know that the broken collagen pathway driven by DDR1 is what builds that barricade, and we know the T cells are standing right outside the gates, knocking on the door via CD96.
What happens if we combine our tools?
Exactly. What if we use DDR1 blockers to cut the power and tear down the walls, while simultaneously using modern immunotherapy to unleash those waiting T cells? Could we turn these incredibly rare, untouchable cold tumors into highly curable hot ones?
It's the exact kind of biological synergy that will drive the next generation of clinical trials.
It turns out that smooth, gray, unfixable machine wasn't featureless at all. We just had to stop looking at the exterior and start reading the code inside.
Beautifully said.
Thanks for diving deep with us today. Until next time.